complete anatomy of the eye. So you are not required to learn anatomy of the eye. But just for the understanding, we need a little bit of basic knowledge of the eye. And this has been learned last week. The sclera, choroid, and the retina. There are 3 layers. The white color protective layer is sclera. Then the blood vessels and the pigmented layer is choroid. And then the retinal layer. Now I am going to draw some of the rod cells in this place. So this represents one rod cell. And another one. And another one. Right. So these are the photoreceptors rod cell, right? So I write here rod cells. Some millions of rod cells sitting on the choroid. Now in front of rod cell, there is another cell layer, and this cell layer is called, they are specialized neurons. So rod cells are photoreceptors. Rod cells are photoreceptors, and in front of that, the second cell is called neuron cells, bipolar cells. So they are called bipolar cells, and they are sitting in front of the rod cell, for example, like this. So bipolar cells, number of bipolar cells are less than rod cells, millions of rod cells, and there is a very less number of bipolar cells, because one bipolar cells can synapse with several rod cells. I just draw another one. And in front of bipolar cells, there is another type of nerve cells. So they are number lesser than the bipolar cells. Now here it represents example 4 rod cells, 2 bipolar cells, and 1 ganglion cell. So from here to here, we can write it's a retinal layer. The retina has then 3 types of cells: rod cells, bipolar cells, and ganglion cells. Rod cells, bipolar cells, ganglion cells. This yellow line represents the direction of the light. So when light energy fall onto the rod cell can convert light energy into a electrical impulse. After that, the direction of signal represented by this white line, and direction of signals this way. That means the nerve impulse, finally exit from the ganglion cell. It's very important to understand when the light fall onto the rod cell convert into a electrical nerve impulse, then it passed to the bipolar cell, then it passed to the ganglion cell. Ganglion cell finally merge with the optic nerve. So optic nerve move into the brain. So many ganglion cells merge to form the optic nerve, and the signal is passing through the optic nerve. So this is the pathway of connecting the receptors of the eye with the brain. In the next lesson, we're going to learn how this is actually happening, how actually the light energy passing to the brain. How does brain know that light is falling to the rod cells? Okay, a little bit complicated lesson. We are coming to understand that. So we'll move to the complete pathway of this cycle, right? Okay, to understand it, first we look how the pigment of rod cell absorb the light. This is the pigment, the chemicals which can absorb light. And this chemical is called rhodopsin. And this represent the light stimulus. The rhodopsin consists of two types of molecules. What are the two types of molecules? Number one, opsin. First molecule is opsin, and the second molecule, this one, cis retinal. Opsin and cis retinal. Rhodopsin consists of opsin and cis retinal. So what is the effect of light? The effect of light has ability to change the bonds of this rhodopsin. So light energy can change the chemical bonds of rhodopsin, because light is an energy form. As long as we are loading energy to the rhodopsin molecule breaks some of the bonds or change some of the bonds. So this change we can visualize like this way. This is opsin, and this one now become transretinal. So what is the difference? The main difference is this cis form become trans. When the cis form become trans, you can also notice in this picture, they are separating. They are no longer together. They separate between each other. And this effect is called a bleaching effect. Cis-retinal converts into trans-retinal. Right, then what is the next effect? To understand the next effect, we want to see exactly where does this happening in the rod cell. Now, when we draw the rod cell, I tend to draw like this, and this is the space for nucleus. And I showed this kind of a membrane folding. And after that, front side synapse with the bipolar neuron. Now these pigments located in this folded membrane, right? So rhodopsin are located on the cell membrane. Right. After that, we have a two molecule. One is opsin, other one is transretinal. After that, there is a complicated series of events take place. So we are not going to learn every single step done by this opsin. Mainly the opsin molecule doing this job. So what we do, we try to summarize all the event in one word, cascade of event. There is a cascade of event. Now, what is a cascade of event? If you can remember the blood clotting, it is an example of cascade of event, right? Thromboplastin release, thromboplastin convert prothrombin into thrombin, then thrombin convert fibrinogen into fibrin, then the fibrin polymerize to make the mesh line. It's a cascade of event. Right. Next, what is the outcome of the cascade of event? So we try to remember the basic steps. Rhodopsin separate into cis to trans retinal bridging effect, and then opsin and transretinal form. Those molecules create cascade of event. Right. The final effect of cascade of event we can summarize like this. So we take the lesson about this membrane potential, resting membrane potential, or maybe the action potential. So if this is a membrane, we have learned previously there is a transporter for potassium, transporter for sodium ion, and sodium potassium pump. So this is responsible to move potassium out, and this is responsible to move sodium in, and sodium potassium pump takes sodium out and potassium in. As a result of that, the membrane can establish resting membrane potential, which is minus 70 milliwatt. Now this cascade of event responsible to close the sodium channel. So this let's say inhibit sodium channel. Right. What will happen if sodium channel is inhibited? The potassium ion is moving out. Potassium ion moving out means the positive charges are going out. Okay. So we have learned previously, inside is negatively charged, outside is positively charged. The membrane inside positive, inside negative, outside positive. Now, potassium ion going out further lose positive charge. Inside lose positive charges because positive charges are going out. And sodium ion brings some positive charges, but it also now not happening. Sodium ion are not bringing any more positive charges because this channel is closed. But positive charges are losing by potassium, and sodium doesn't bring any more positive charges. So what will happen now, if we try to understand, there is a value for, there's a value for inside, let's say it maintains certain value like a minus 40 milliwatt, example. If this rod cell maintain minus 40 milliwatt, now with this thing happen, what is going to happen to this minus 40 milliwatt? It's getting more negative because inside is getting more negative. So when it get to more negative, we could expect this is like even minus 60 milliwatt. It can reach to minus 60 milliwatt. So this cause hyperpolarization. Getting more negative is hyperpolarization, right? So let's summarize again the four steps, starting from here. Light hit the rhodopsin, then the rhodopsin changed to an opsin and a transretinal. Then there's a cascade of event. The cascade of event inhibit sodium channel. This lead membrane hyperpolarization. Membrane become hyperpolarization. Okay, so we are coming to the next level then. In the next level, you can find the hyperpolarization cause to make a generator potential. Hyperpolarization cause to make a generator potential. Now think whether you understand this. It is getting more negative. This more negative situation, it create kind of action potential. Right. Uh, if you look at the textbook mention same way, the hyperpolarization cause generator potential. But what we have learned so far, this is not possible because we learn opposite of that. That means negative minus like the minus 70 is the resting membrane potential and the minus 70 milliwatt first going close to minus 55 threshold level. That threshold level continues to fire up to plus 30 milliwatt. The plus 30 milliwatt is the action potential. So it should change from minus 70 to plus 30 to generate action potential. Now, if you really think that is the real base concept of action potential, how do you understand now when it's going to opposite direction minus 60 milliwatt, it can make a generator potential or action potential. So we call generator potential based on the individual cells. One rod cell can create a generator potential. Finally, these potential merge retinal convergence to make a one action potential. Right. I think textbook doesn't mention anything. It is go on like this. This generator potential cause nerve impulse, the optic nerve, and then communicate to the brain, and then brain aware the light is falling to the red cell. So light is falling to the red cell is a message conveyed by the brain. Now here, coming back to again to this picture, direction of light is this. When the light fall onto the rod cell, there is a action potential, generated potential created by each cell, and then optic nerve communicate to the brain, and brain identify the light. Okay, now we will think in two different ways. One is, if you are asked a question in the exam and you have to write answer for the question, you need to write the basic steps. The light absorbed by the rhodopsin, then cisretinal convert into transretinal, then opsin and transretinal separate. After separating opsin and transretinal, it creates a cascade of event. This cascade of event closing the sodium channel. Sodium channel closing lead inside getting more negative called hyperpolarization. Then the hyperpolarization cause to make a generator potential. This generator potential communicate via optic nerve to the brain. And so the last step maybe action potential, and this cause brain identify light. So this is one aspect. We can go on like this and finish it off like this way. So this has been completed now. To complete the cycle identification of light by the rod cell is like this way. Only the doubt you have now, how can hyperpolarization make a generator potential? Right. You don't want to know this one, you don't want to learn this one because it is not important to get the marks. But I briefly outline what is exactly happening. If this is a rod cell and this rod cell synapse with bipolar neurons, right? So right. So we try to understand it like this. Let's first assume there is no light. If there is no light, the rhodopsines not separate and sodium channel is not closed. So when sodium channel is not closed, there is a certain limit of membrane potentials. As we learned before, we will say it is minus 40 milliwatt. It maintains this when in the absence of light. And this creates a continuous action potential of rod cell with the brain. So these highlighted cells continuously sending impulses to brain, and brain identify it is as dark now. So I hope you understand now. It can make a continuous signal to the brain when it is dark. That means synaptic transmission takes place. Now when light comes, we said the final effect is hyperpolarization. Just go to minus 60 milliwatt. The hyperpolarization is actually stop the nerve impulses, right? So stop action potential. So now there is no action potential from this place. Then what will happen? There is a different bipolar neurons activate. For example, this bipolar neuron activate. The yellow side is closed due to hyperpolarization, and the other side activates. So now this one communicate with the brain